Hello, welcome to Biology, the scientific study of life. Asexual reproduction. In addition to sexual reproduction, many fungi can reproduce asexually. Colonies are produced by mitotic production of spores, which can be spread by air or water. Some species only reproduce asexually, as with sexual reproduction. The process of asexual reproduction vary widely among fungi. Some fungi that can reproduce asexually grow as mold. Notice that. Mold is a familiar sight in the kitchen where it forms foreign carpets in fruit, bread, and other foods. Molds grow rapidly as mycelia and produce spores. Many species that can grow as molds can also reproduce sexually if they come in contact with other mating types. Other asexual fungi are yeasts. Yeasts inhabit liquid or moist habitats, including plant sap and animal tissues. Notice that. Other sexual fungi are yeasts. Instead of producing spores, yeasts reproduce asexually by simple cell division or by the pinching of small bud cells of a parent cell. Some yeast species can also grow as filamentous mycelia, depending on the availability of nutrients. Yeasts sometimes can also reproduce sexually. Many molds and yeasts have no known sexual stage. Biologists who study fungi or mycologists have traditionally called such fungi ditromycetes. The suffix mycete means fungi or imperfect fungi, from the perfect refer to sexual stages of life cycles. Whenever a sexual stage of a so-called ditromycete or mycete discovered, the species is classified in a particular phylum, depending on the type of sexual structure it forms. In addition to searching for little-known sexual stages for such an assigned fungi, scientists also can now avail themselves of genetic techniques to identify taxonomic status. Fungi descended from an aquatic single-celled flagellated protists. Data from both paleontology and molecular systematics offer insights into the early evolution of fungi. Systematists now recognize fungi and animals as sister kingdoms. In other words, fungi and animals are more closely related to each other than they are to plants or other eukaryotes. The origin of fungi, phylogenetic systematics suggests that fungi evolved from a flagellated ancestor, while the majority of fungi lack flagella. The lineage of fungi thought to be the earliest to diverge. Moreover, most of the protists that share a close common ancestor with animals and fungi also have flagella. These three groups of eukaryotes, fungi, animals, and the Protestant relatives. Phylogenetic evidence also suggests that the ancestor of fungi was unicellular. The fact that other evidence indicates that animals are more closely related to some unicellular than to fungi suggests that animals and fungi must have evolved multicellularity independently from different single-celled ancestors. Based on molecular clock analysis, scientists have estimated that the ancestors of animals and fungi diverged into separate lineages 1.5 billion years ago. However, the oldest and disputed fossils of fungi are only about 460 million years old. One possible explanation for this discrepancy is that the microscopic ancestors of terrestrial fungi fossilized poorly. This is the fossil fungi. The move to land. Much of the fungi diversity we observe today may have had its phylogenetic origin in an adaptive radiation when life began to colonize land. Fossils of the earliest known vascular plants from the late Silurian period contain evidence of mycorrhiza, the symbiotic relationship between plants and subterranean fungi. Plants probably existed in this symbiotic relationship from the earliest periods of colonization of land. Fungi have radiated into a diverse set of lineages. The phylogeny of fungi is currently the object of much research. In the past decade, the molecular analysis has helped clarify the evolutionary relationships between fungi groups, although there are still areas of uncertainty. Presents a simplified version of a current hypothesis of fungi phylogeny. Let's talk about chytrids. Chytrids, this is the classification of chytrids. This is this one. Et voila. Fungi classified in the phylum Chytridiomycota, called chytri or chytri. Chytrids are ubiquitous in lakes and soil. Some are several, others parasitize protists, plants, or animals. Molecular evidence supports the hypothesis that chytrids diverged earliest in fungi evolution. Like other fungi, chytrids have cell walls made of chitin, and they also share certain key enzymes and metabolic pathways with other fungi groups. Some chytrids form colonies with phyla, while others exist as a single spherical cells. But chytrids are unique among fungi in having flagellated spores called zoospores. Until recently, systematists thought that fungi lost flagella only once in their history, after chytrids have diverged from other lineages. However, molecular data indicates that some chytrids are actually more closely related to another fungi group, the zygomycetes. If this is true, then flagella were lost on more than one occasion during the evolution of fungi. Notice this multiple evolution losses of flagella. For this reason, many systematists consider the phyla Chytridiomycota and Zygomycota to be paraphyletic. Notice that this is chytrids. Notice that. Closer look. Zygomycetes. The approximately 1,000 known species of zygomyces fungi in the phylum Zygomycota exhibit considerable diversity of life histories. This phylum includes fast-growing molds responsible for rooting produce such as peaches, strawberries, and sweet potatoes during storage. Other zygomycetes live in a parasite or as commensal neutral symbionts of animals.